Good evening, everyone. I'm just going to give a few more minutes for the attendees who are still joining to settle in, and then we will start off with introductions. Please just bear with us for a few minutes. Thank you to everyone who has taken the time to attend our annual CPD event. Tonight, we are joined by Dr. Neil Spooner, who is the co-founder, chair, and director of the Patient-Centric Sampling Interest Group. It is a non-for-profit organization that brings together a variety of interest parties who wish to develop and promote the use of patient-centric sampling technologies for blood, plasma, and other human matrices to better facilitate the advancement of human healthcare and well-being. Neil is also the founder and director of Spooner Bioanalytical Solutions, a consultancy based in Hertford, UK. And in addition to this, Neil is a senior visiting research fellow at the School of Life and Medicine Science at University of Hertfordshire in the UK, and also the editor-in-chief of Bioanalysis Journal and the chair of the Read Bioanalytical Forum. He has published extensively with over 70 peer-reviewed manuscripts and more than 50 podium presentations at international conferences and symposia. Neil's current focus is on working with companies to help them understand the benefits and challenges of microsampling for the generation of high-quality bioanalytical data and helping them integrate the technologies into their pre-analytical, clinical, and bioanalytical workflows. Further, he is assisting innovator companies to understand their customers' requirements for microsample collection, processing, and analysis technologies, enabling them to develop and market devices supported by high-quality scientific data. Prior to all of this, Neil has developed extensive experience in the quantitative bioanalysis of drugs, metabolites, and biomarkers in the pharmaceutical industry and contract research organizations in both the UK and in the US. In over 20 years of industrial practice, he has led groups operating in the discovery and regulated arenas of clinical and preclinical quantitative bioanalysis and metabolite identification. Neil has extensive experience and successful leading international departmental and cross-functional initiatives, including implementation of new technologies and workflows. And in his private life, he's also a proud husband and father of two daughters. In his spare time, he enjoys traveling with his family, playing guitar, skiing, walking the dog, and is all over just a really pleasant person to know. Neil, thank you very much for taking the time to talk to us today, and we really look forward to learning about microsampling.
Thank you, Elnie. Thank you so much for that. And thank you, Elnie and Chris, for the invitation today. I'm very excited to be talking to you all and looking forward to your questions at the end and seeing what activities might arise out of this. So, the title of my talk is Introducing Patient-Centric Sampling, what it is and how it could transform clinical diagnostics. And hopefully by the end of the presentation, you'll understand a little bit what we mean by the term patient-centric sampling. So let me just make sure I can change my slides. So what is patient-centric sampling? Um, some people like to call it microsampling, but it's not just about collecting smaller volumes of blood. What it is about is thinking about how we sample human beings, not just for blood, but for other biological fluids, that puts the needs of the patient first. For whatever reason, the technologies we have been established for a long time because they work, but basically they put the needs of the lab, the clinicians, a lot of people first, but the patient seems to come last. And we take blood from people in a way that a lot of human beings don't like. And more importantly, a lot of the blood volume we collect, a typical blood volume might be 2 mils or more. Most of our analytical techniques now only need a few microliters. So most of that precious lifeblood is thrown in a waste, which I personally think is unethical, particularly now that there are other approaches that give alternatives. And the alternatives I'm going to talk to you about today aren't necessarily meant as replacements for traditional venipuncture. They're there as an alternative. And as we'll talk through, there are maybe places and certain population groups and patient groups that would benefit from collecting samples in this way. Maybe they're having trouble collecting samples at the moment. And it basically hopefully gives patients and healthcare systems a little bit more choice and doesn't rely on a centralized system of traditional phlebotomy. So um, it is about collecting an appropriate sample using a process that's most convenient for the patient. We want to provide high quality information. We don't want to lower the barrier on quality at all. And because we still want to make high quality decisions in clinical. But the technologies I'm going to show you don't necessarily involve tiny volumes. Some of them are still reasonably large volumes. So that's why we don't like to use the term microsampling. We prefer to use the term patient centric sampling. So, what are the benefits of shifting to patient centric blood sampling? Um, Putting the patient first, reducing the impact of sampling on the patient. So, minimizing pain and invasiveness. We know a lot of people really don't like needles. Um, we're facilitating convenience, potentially sampling at the home or somewhere that's more convenient for the patient rather than them having to take time out of their day to go to a clinical facility just to have a blood sample collection. And as I said earlier, we're looking at optimizing the blood volume sampled. So take the volume of blood you actually need to perform the tests, not taking a volume of blood just because that's the size of the tube you have. We have possibility of enriching data here because we're able to collect samples in different times and different locations. Maybe we can monitor our populations and whether they're well people or sick people, we can monitor them more often and hopefully monitor them before they are actually sick. Um, but quality is always key. So these technologies I'll show you still obtain high quality blood, plasma, and serum sample to do clinical diagnostics. So what kind of groups might this benefit? So we know a lot of vulnerable populations maybe don't work well with standard venipuncture approaches. And we know that pediatrics, small children often don't like having blood taken. It's difficult sometimes to get the volume that you need, and sometimes there are ethical reasons for not taking large blood volumes. Elderly can be difficult. I know with my own parents, blood sampling can be a big problem and can cause a lot of distress, a lot of bruising, difficulty finding veins. Not a nice experience, not something that certainly my father wants to repeat on a regular basis. Um, Oncology patients may have difficulty giving blood. Anemic patients, why are we taking so much blood off people who don't have many red blood cells? And we've seen some great examples recently here in the UK and elsewhere of patients with learning difficulties who, for whatever reason, find it difficult to have a blood sample taken and potentially have either never had a blood sample taken or haven't had a blood sample taken for years. Despite all sorts of therapies to help them have blood samples taken, it still becomes a stressful occasion. And we've got some videos on our websites of communities that have really, with learning difficulties, that really benefited from this and now find it a positive experience having a blood sample taken rather than a major negative experience. Obviously, we've got patients with fragile veins, patients with needle phobia, and also importantly, patients in rural locations. If their nearest phlebotomist is several miles away, that can mean a large amount of their day being lost to just collecting the blood samples. And obviously, modern individuals are busy. Parents, children, got busy lives. Fitting in clinical appointments just to collect the blood sample can be difficult. So why not enable them to collect the sample in the comfort of their own home in the evening when it works for them, or in the morning when it works for them, or even when they're on holiday. So we open out the options. As I say, Standard phlebotomy isn't going to go away. This is just going to bring some alternatives. So this slide just shows you some of the many devices that are available commercially on the market. There's more than this available. Um, Towards the end of the talk, I'll put up some QR codes that help you go to our website and our LinkedIn group. And on our website, you can find listings of many, many commercially available different devices that you might be interested in. So this is just some of them here, and I'm going to go into some of the devices and what they look like, what the benefits are, and what the challenges are to implementing these in the next few slides. So a number of you may be familiar with patient-centric sampling and microsampling from where it all started really with dry blood spot sampling, which came out of neonatal screening over 60 years ago. Um, so dry blood spot samples are very well established technology, particularly for screening. Um, they use a finger stick to collect the blood sample. They're very cost effective. They're made of cellulose, paper, and card. Um, so reasonably cheap to buy in large amounts. Um, and they can work well with children, elderly, remote areas, and home sampling. The advantage of the dried sample is often that the analyte is stabilized in the dried sample, so shipping them can be a little bit cheaper if you've shown that stability. Um, so you're dealing with whole blood here, so you don't need to centrifuge the sample. No need for dry ice and freezers, and there's no plastics involved. So a bunch of advantages to dry blood samples. However, in order for quantitative analysis to be performed, you need to be able to spot an accurate volume or punch from the sample. So you need to either put an accurate volume onto the card, which is a difficult thing to ask a patient to do. And believe us, many people have tried doing that. Or you need to take an accurate diameter uh, punch, sub punch from that sample. Um, there's obviously issues with compatible with existing analytical workflows. The volume is much smaller. We're talking a blood sample rather than plasma or serum, and it's a dried sample, whereas most of our analytical flow workflows work with liquid samples. Um, and obviously, all the reference ranges may be wrong because we're dealing with blood rather than plasma or serum. But there's a bigger problem with traditional dried blood spots. It works fine for neonatal screening, but when it comes to actual clinical chemistry or measuring accurate numbers with good precision and accuracy, we can't rely on this technology to give us good data. And one of the big problems is around blood hematocrit. So the amount of red blood cells in the volume of blood. And this can vary between patients, can vary time of day, can vary between the sexes. And as you can see here on this illustration, we've spotted they've each spotted a fixed volume of blood here with 20% hematocrit at the left, 45% hematocrit in the middle, and 70% hematocrit in the right. And then taking a fixed diameter punch, you obviously get a different amount of the blood spot. So we're expressing our concentration values as units per mil of blood. And if we're out of control with the mil of blood, we can't give very accurate data. And we know that this can give error values of up to 30%. So maybe when we're making truly quantitative assessments, it's not a good approach. But thankfully, and I'm going to talk you through, there are some alternatives that are useful. So if we're wanting to stick with the dried blood sample, so stick with those advantages of very small volumes, being able to potentially ship at room temperature if you demonstrated stability, there's a number of options available. The main ones being the Neoterix Mitra device and the Capitainer B cards using fingerstick, and the Tasso device, which you can place on the upper arm, press a button, and it pierces the skin and a couple of minutes later you've got a sample. So the thing about these technologies is regardless of the blood hematocrit, they will always collect a fixed volume of blood. So you get rid of that hematocrit problem, you get rid of that requirement for the patient to have to spot a fixed volume of blood, and we get a high quality sample with a fixed volume. Um, the problem is they're obviously far more expensive than a dry blood spot card or a standard blood tube. And a lot of them contain a lot of plastics as well, which can be a lot of issues with disposal and recycling. So there are some options out there for having the advantages of dried samples, but being able to produce a plasma-like material. I won't call it plasma because it's not produced by centrifugation. But basically, these devices are able to remove the red blood cells and leave behind a matrix, but they remove other components as well. So the matrix they produce is not the same as plasma, but a lot of people also like to use these technologies as well, and they're an interesting alternative. But if we want liquid blood, there's a number of options out there. Again, I've just highlighted a couple of options here. The Winos device, um, which applies a slight vacuum to a finger stick to collect blood into a microtainer. Griner and a number of the other tube manufacturers sell tubes now that are optimized for helping you to collect a blood sample from a finger prick. However, still the tube types and the sample volumes may not always be compatible with existing workflows. Um, we've got a capillary blood sample rather than a venous blood sample, and that may mean that the reference ranges again could be a little bit different. We're dealing with whole blood, and potentially in an ideal world, we don't want to freeze this, so we need to assess the stability, and some analytes may not be stable. I've not got it in listed here, but another issue can be with hemolysis with these kinds of samples, so that may affect some assays as well. And it can be difficult to get large volumes from finger stick. What we find is um, some patients like the idea on paper of a finger stick and collecting a reasonably large volume up to 500 microliters from a finger stick, but we find that not many of those patients come back to do it a second time. They maybe not don't like the finger stick process, or they may not like the process in order to collect the larger volumes required. But it is an option, and the options I'm going to talk you through, different solutions might work for different patient groups, for different analyte types. There's no single set solution. Um, One of the areas that is very exciting is these devices that can be placed on the upper arm. They can also be placed on the thigh, the back, the buttock, anywhere that's reasonably well vascularized. But we find that the nerve endings are not as many as they are on a finger. And these devices all involve just placing these devices on your arm, maybe after a little bit of warming, pressing a button, and after 2 or 3 minutes, you have a blood volume of, depending on the device, between 500 microliters and 2 mils. And in fact, that Let's Get Checked device works in a way that you're able to change the tubes on there. So if you require multiple tubes for different assays with different anticoagulants or different stabilizing agents, you can change the tubes on that. So we can collect larger volumes of blood and less painful for many people. And it's a lot of these devices that are out there being used with elderly and people with learning disabilities and people doing therapeutic drug monitoring, etc. And obviously, as you can see, potentially the cost of these devices is higher, although we can argue if you do a cost benefit analysis, the fact that the person is not taking time out of work or school or their life to collect a sample, the reduced need for phlebotomists, etc. Um, there's been a number of publications on cost benefits, and for a number of disease states, it actually works out even with the higher cost of consumables, that these can be very comparable, if not even cheaper, than traditional phlebotomy when you take all expenses into account. Big problem is the amount of plastics, disposal, and environmental impact. Um, so, what analytes can I measure using patient-centric sampling? I'm not listing all of them. My background is not as a clinical chemist, so please apologize if I get some of this a little bit wrong. But these technologies have been used for infectious disease screening, HbA1c for diabetes glucose control, although not the measurement of glucose itself, neonatal screening, antibody screening. We saw a lot of it during the COVID epidemic. People are measuring metabolic panels, lipid panels, liver function panels, hormones, vitamins, cancer markers, and a lot of prescribed drugs that therapeutic drug monitoring is required for, things like anti-rejection therapies for organ transplant patients are commonly used that have narrow therapeutic indices. So it all sounds very rosy offering these new options, but obviously it's not all plain sailing. So what are the limitations? Well, obviously, we're now putting the collection of samples into the hands of the patients. So that changes a lot of things. We've got the potential for failed collections. Some of the technologies are quite good at indicating that you've made a good collection. The sample volumes are smaller, so if you've not moved to more modern instrumentation that can take smaller volumes, that might be a challenge. We've obviously got complex logistics, so rather than just having samples being collected from a number of clinical hubs and sent to their local lab, we now have to get consumables to people's homes or a local pharmacy or health centre, and we then have to get those samples back to the analytical laboratory. So that's a little bit more complex than we have at the moment. We have to prove who the patient was. You know, we've seen in clinical trials that people weren't on that day weren't keen on collecting the blood sample, and for whatever reason, their spouse collected the blood sample. I'm not quite sure how that was going to help with the clinical trial or with their healthcare, but this is the kind of crazy thing that human beings do. It's very important to get involved with patient training. There's suddenly being given a whole pack that they've got to collect samples themselves. We tend to find that it works best if the first time people use these technologies, it's done in a clinical setting with training by a healthcare professional. That tends to result in higher percentage getting it right first time subsequent to that. These technologies are not currently useful for measuring things like glucose, blood gases, potassium and calcium. Not good for blood cultures, coagulation studies, ESRs, and cross matching. Some of these tests require either they've got higher variability or require larger volumes, high precision, or potentially affected by tissue fluid contamination. But there's a lot of work being done on expanding the list of analytes that are available. So challenges elsewhere. So people that work in the analytical lab, we know for experience, we get a mixed bag of people reacting to these technologies. Some think this is wonderful, this is good for the patient, we should be embracing this. Yes, it's a different workflow for us in the lab, but we see the bigger picture. Whereas we get other labs that say, oh my goodness, I've got samples in a different format in appropriate volumes, and maybe it's blood rather than serum. I really don't want to deal with this kind of change. I can't afford the time to qualify the methods, and we know that kind of reluctance to change, and that's completely understandable. But on top of those, normal human responses, we have to think about what our methods look like when we're validating the methods and qualifying the methods. So if we're now wanting to ship our samples at ambient temperatures, what does that mean? You know, that could be very high temperatures, that could be very low temperatures for elongated periods of time. So we need to reflect this in our validation. And some compounds, some analytes will not be stable and may need um, something. There's a number of companies now selling devices that will help maintain the temperature of a liquid microsample while it is shipped. Um, there's also, in the early days of some of these technologies, there were differences in lot to lot variability. So you needed, if you were wanted to go on through a clinical trial or healthcare, you just need to make sure that as you buy a new batch of these devices or these samplers, that you're not getting different numbers. Um, we know that for dried samples, hematocrit, while we overcome the issue, the volumetric issues talked about before, for some analytes, it can affect the recovery of that analyte with the variability of hematocrit. But there's things you can do in the extraction that minimizes that impact. You just need to spend a little bit of time looking at that. Also, with dried samples, you're not adding an anticoagulant, whereas your quality control samples and standards will use blood that has anticoagulants. So you need to assess whether that's going to make a difference. And particularly with dried samples, none of these things really affect the liquid samples. How do you do dilution with the dried samples? And there's way to do that, but it's just a little bit more complicated, a bit more for people to get their head around. Um, and obviously, the big one is, are these different formats and sample types compatible with existing lab workflows and automation? People don't want to be spending a lot of money retooling their labs, retraining their staff. And obviously, there's big concerns about the quality of the collected sample. Have you got the correct patient? Has the sample been hemolyzed? Have you got enough volume? So, other challenges in the clinical area, if we're collecting now whole blood, um, there's the option with the liquid blood to be able to centrifuge that at the lab when it arrives and get serum or plasma. So we don't have comparison there other than the concern of stability during shipping whole blood. But you know, with the dried samples, or if you're not willing to centrifuge your samples, um, the reference intervals and decision limits could be different for capillary and venous samples, whole blood versus plasma and serum, and wet versus dry samples. Um, we talked about before the logistics. How does that work? Um, and the quality of the collected sample is always going to be a concern. And you know, what's the cost like versus the benefit? And each individual use case in each region has to assess that for themselves. So some of the biggest challenges to roll out really are the logistics. How do you confirm the patient identity when they collected it themselves? And there's a lot of smart things going on with apps on phones to help do that. How do you ensure continuity of supply? You know, a lot of these technologies are relatively new companies. Are they going to keep on producing these technologies into the future? Um, Labeling can be an issue. Some of these technologies don't give as big an area to attach the same kind of label that people are used to. What are the expiry dates of the actual devices themselves? Um, you know, it may be that you need to buy in a large batch. Are you going to be able to use them several months, several years later? And how do you store these devices prior to use, particularly if you're sending them into people's homes and their locations where they live? Are you putting a burden on them to safely and securely store these devices? Um, we've obviously got concerns about sample integrity and chain of custody. What happens moving these samples from the home to the analytical lab? There's ways of monitoring the samples, of stabilizing the samples, but it's just something we all need to think about. And are the devices available in the different countries? Are they approved in the different countries? We already know that different devices are not necessarily regulatory approved in different countries or even commercially available. And obviously, we've got a training burden for the staff involved, the patients, or if the patients are unable to collect their own sample, their caregiver. And obviously, this needs to take into consideration the multiplicity of languages that may be in the patient population. Um, and obviously, there are challenges for the regulators and the people paying the money for the tests. So, different country and regional differences in regulatory status of these devices, acceptability of the data. Um, are we happy with the patient reported data? Are we happy if sampling time is important? Are we confident that we have the right number there? As we said before, patient identity can be important, cost benefit, and sustainability. We're always interested in what happens to these devices when they go into the disposal chain. So, where do we go to next? Well, with regards to the African community, I'm really excited to let you know that Patient-Centric Sampling Interest Group have been working with the African Society for Laboratory Medicine, and we'll be presenting two ECHO webinars focusing on opportunities for patient-centric sampling in African diagnostics. The first event is on Thursday, the 23rd of April at 4 p.m. East Africa time. And we have a number of speakers there. We have an introduction to existing patient-centric technologies from Jolene White. And then we have two case studies taken, one from Chris Hendricks and one from Makobo and Rose, that have been actually done in the African environment. And then the second event, we bring together those speakers and a broader panel for a discussion about the potential of patient-centric sampling for Africa, what the potential use cases are for these technologies in Africa, what are the challenges to implementing these technologies, but most importantly, the thing that the patient-centric sampling interest group likes to do is figuring out ways to address these challenges. Any new technology, any new approach is always difficult, and some people like to shy away from that, and some people like to embrace that. Both are correct, but we like to figure out ways that we can overcome these challenges, and we like to build action plans. And in this particular case, we very much like to help support if the community are interested in these technologies. We'd like to very much um, support ASLM and other regional groups that want to move this forward, and PCSIG play a role in helping to support that and move it forwards and building action plans to actually do something. So for those of you that are interested in what we're talking about here, you might like to get involved with the Patient-Centric Sampling Interest Group. We're a not-for-profit group, as Alna said at the beginning. We have a lot of resources on our website. If you've got your camera, just hold it up to that QR code on the left-hand side, and that will take you to our website. The QR code on the right-hand side takes you to our LinkedIn group, where we have a community approaching 700 people that share information, help each other, ask questions, answer questions, share publications, etc. Or just contact us at contact@pcsig.org, or my email address is Neil, Neil, at pcsig.org. And the kind of things that you might be interested from our website is we run webinars every month. Earlier today, we ran one of our latest webinars, which was about PKU analysis and some of the issues using traditional dried blood spots there. Um, we have on our website all of our webinars. I think there's 32 or 33 of them now, of different use cases for these technologies from different groups around the world. They're also all available on our YouTube channel. We have listings of all the commercially available samplers and a variety of other technologies. We have a library that will help you find maybe some publications that might be of interest. There's resources for clinical trials. There's resources for a lot of different things on our website. So I encourage you to take a look at that. And but if you have any questions, please come and find us. So the conclusion to this talk is there's a number of benefits of patient-centric sampling for clinical diagnostics. We are seeing increasing interest and acceptance of these technologies. We're starting to see them being introduced into healthcare in a number of regions and countries. However, as we talked, there are a number of challenges to routine implementation, and these challenges extend beyond the laboratory. But, we're here to overcome these challenges, and we're a big believer in working together, finding passionate, persistent people to help us move this forward. I'm very excited about the conversation to come. So I'd just like to say thank you for inviting me to speak today. Hopefully you find it interesting. Hopefully we've got a little bit of time for questions and discussion. Just if you are interested in further reading, there's this book which I edited a couple of years ago, Patient-Centric Blood Sampling and Quantitative Analysis. It has a number of articles that might be of interest. And also, I'll leave that up there for a moment if people want to take a photo of the screen. There's a number of publications there that might be of interest to people, but there's many more on our website. And if you're interested, as I say, join our LinkedIn group. So, no further ado, I'm open for questions.
Neil, thank you. Thank you for an insightful presentation.
Should I stop
I
sharing,
think,
Chris, so I can see you?
yeah, sure.
Okay.
I think while people are gathering their thoughts, maybe I can start off with one or two questions. I think I was the first person to import the Neoterix device to South Africa. It was just part of
Oh,
a research project, and it was very expensive.
yes.
And then one of the suppliers, I think they must have picked up that I ordered this because then they got the distribution rights in South Africa, and then the price even doubled.
Oh,
And the collection price turned out to be far more expensive than most of the tests that we would do on it. So, I think that for me is a great hurdle to the implementation of these devices. That's the one thing. And then the other thing is, I mean, if you go to these pathology laboratories, they've made a huge investment in automation using the traditional vacutainers. And unless you've got some kind of a front end process by which you can ensure that these patient-centric collection devices can enter the automation pipeline seamlessly, I think it would be a difficult market to crack.
yeah.
So, maybe it's part of a question, part of a statement. Maybe one should look at, the kind of opportunities where these factors are not that important. So perhaps somewhat lower volume testing, and then also testing that's in any way has some manual steps involved. It's like typically specialized testing and so on. It's like, for example, I think drug monitoring is also a good example. These may be good entry points for these kind of collection devices. I don't know what your thoughts is on that.
No, I agree absolutely. That start with places that you've got the best chance of success. So if you've got an alternative workflow that this would fit into, then why not go for that? If you've got a patient group that will be particularly benefit from this, that you could take on board the added complexity. But what I would say is, I think the power of hopefully talking to you and having the echo webinars that are coming up is hopefully different laboratories can come together and lobby the manufacturers over the price, lobby the manufacturers over integration into existing workflows. We now know that a number of for the liquid samples, a number of the tubes that these technologies use are now integrated into the auto analyzers everyone is buying. So we're addressing that. But if there are particular concerns in your laboratories, I think this is where the power of patient-centric sampling interest group comes, because we try and pull together people from different organizations, from different backgrounds, who tell us this is our problem. This is our challenge. And okay, let's find a solution. And if the problem is price or fitting into automation, let's talk about that. And let's engage with the manufacturers of either the devices or the automation and have that conversation. Because if we're able to say these technologies will bring these benefits to our healthcare systems, to our human beings in our region and our country, we want to do this, but we need you to help us on the price point. And, I'm a firm believer that also, as we open this up, um, there will be new innovations come along to fit this in. But yes, I agree with you also. Initially, just taking the low hanging fruit, finding the analytes, the assays, the patient groups that this best fits for, and let's get some success stories out there and then build on that.
Yeah, I think, I mean, the dried samples have got obvious advantages in South Africa and Africa. Sample stability is a major issue, and we're dealing with some very harsh temperatures and so on. So, if one could actually get away from those tubes, I think it would be very beneficial. And probably, I mean, there's quite a few labs that is already working on dried blood spots, besides the newborn screening laboratory. So, I mean, in South Africa, HIV is often screened for on dried blood spots and so on. So, if you can extend that approach and the automation that's already there to some other analytes, then, that could be, it could be an entry point. The dried blood spots obviously is still very popular. What about the idea of normalizing for hemoglobin content on the dried blood spots? Is that something that has been investigated?
So I think it was LabCorp from the States did a publication 2 or 3 years ago of normalizing to chloride, I believe it was.
Oh, yeah, I remember. Yeah.
And I think that works quite well and gets around some of the problems. But you've then got two measurements that you've got to make, each with their own variability. But yes, I think rather than normalizing to hemoglobin, I think they chose to be, or other constituents. I think they found that chloride was the best one.
Yeah. There's still no questions from the audience, so I'm just going to continue asking some of
Okay,
my questions.
so they're obviously just stunned.
Yeah. So, I mean, I think globally people are moving away from urine as a sampling matrix. However, we still do quite a few of our metabolic tests in urine. Even something like acylcarnitines, which I think these days are seldomly done in urine. We still do that in urine. And it seems like an obvious target for dried sampling. So if you collect urine on paper, dry it with the necessary preservatives and stabilizers and so on, and that should be a fairly easy entry
Yeah.
point. So I don't know, you got any thoughts or
So
experience with that?
there's been quite a few publications, I believe, on dried urine spots. Yes, and those using the same technologies are available, and you wouldn't need the volumetric approaches. You can use the cheaper approaches. I think we have some of our members are developing more patient-centric approaches to collecting urine, because obviously urine is not necessarily for some patients is not the easiest sample to collect in a cleanly and a manner that we would all like to do. And so what would the steps be involved in collecting a dried urine spot? Would you just pee onto a collection stick? Would you pee into a collection vessel and then transfer some? That needs to be thought through, I think. And again, you need to normalize for another analyte in there because volumetric concerns. But I think it's something that should definitely be investigated. And if that would be useful for some of your populations and patient groups. Uh, why not? Um, it's not being commonly used, but there are publications out there with dried urine.
Yeah, my thinking is, I mean, I think the collection is not easy because you need a midstream sample.
Yeah, yeah.
But my thinking was along the lines of a device that would automatically, discard the first amount of urine
So
and
there,
then collect
there
the
are
second amount.
there are devices out there. So the one that comes to mind is a company called Forte Healthcare, I think it is. It's called the Peasy device. So it automatically it kind of it's designed mainly for females to collect a midstream to minimize contamination, and it automatically discards the first part and collects a midstream sample in a normal sampling tube that then can be detached, capped, and sent in. It's very much less spillage, more assurance that you have got a real midstream, and less contamination. The problem with a lot of midstream urine samples, I believe, is bacterial contamination.
Yeah. So, I mean, if you've got a device like that that automatically just collects into a chamber that contains cellulose or whatever, some matrix that can be used to dry the urine samples, and that seems
So the
like
liquid
a good
version is
solution. I mean, the other
available.
thing is,
I'm not aware of dried versions, Chris, but the liquid
okay,
versions are available.
okay. Yeah, interesting. There's no questions from the audience tonight. That's quite
Okay.
strange.
Well,
But there are
I
some
want to
comments
talk
to
a
say
little.
that people have enjoyed the presentation.
Yeah, hopefully, with regard to the ASLM presentations, um, the way ASLM works with its Echo webinars is it doesn't open registration, I believe, until a few days before the event. So as soon as registration is available for those events, I will post it to our LinkedIn group, and it will be available through that. I was hoping registration would be open by today, and I'd be able to include the details on this presentation. But unfortunately, I talked to them today, and it's not open yet. But it is going ahead. We have our practice session tomorrow, so we're very excited about that and looking forward to that and seeing opening this. All the questions you were asking, Chris, opening those out to a bigger audience across Africa, hopefully, and trying to figure out how this is there value in these technologies for the continent of Africa, and if there is, how do we make it happen?
Mm. Yeah, there must be. It's just how to make it happen.
Yeah, yeah, yeah.
Neil, I've got one or two just out of curiosity. What has been the uptake in the US and the UK with the microsampling devices for diagnostic labs?
I'd say it's patchy. We're reaching a tipping point, but one thing that's just happened in the last few months is the UK government have issued a 10-year plan for the National Health Service. And a big part of that is moving healthcare closer to the patient and being more patient driven. And so there has been a call by the NHS to the manufacturers of these technologies and of the automation instrumentation to give their opinions. And I believe later this year they're going to bring them together in a workshop to work through how this might, what the use cases might be within the National Health Service in the UK to make this standard of care. Otherwise, it's patchy. Different laboratories may have adopted it for different patient groups, and it's gradually spreading. And the Association of Laboratory Medicine ran a conference last year. They're running another conference in September focusing on this. And we're doing the same. So the Patient-Centric Sampling Interest Group are running, if you look on our website and on our LinkedIn, we're running some events in Europe and North America over the next few weeks, looking at what the challenges are to more widespread implementation and how we might overcome that. And we've got speakers from the FDA, the MHRA, etc., joining in that. And so globally, you go to the Netherlands, you go to Belgium, it depends on the local awareness of people, of clinicians and people in the labs, and the local passion of people in the labs and clinicians and patient groups in some cases as well. So we saw this recent case with patients in the northwest of England with learning disabilities that have rarely been sampled, and we helped make a video of that that's available on our website. A young lady with Down syndrome and her mother, and the 35 people that took part in the trial, it transformed their healthcare. And that has now been transferred, that whole region is now moving that to the standard of care for 2,000 patients with learning disabilities. And we'll be looking at how that can be spread throughout the nation. So it's early days, it's still happening. It's complicated.
One other thing I was wondering about, so in an Africa setting, for instance, if a patient uses the Tasso or the any of those type of devices, one of the big challenges are the time it will take the sample to go from collection to the lab. Is there anything built into, I don't want to get too technical, but is there anything built into these devices to make sure that it's more stable and it will overcome a longer
So
delivery time or stuff like that?
it's it's analyte by analyte, as you can imagine. Different analytes are going to be more stable than others, and it's dependent on the environmental conditions found. But there are a number of companies associated with the patient-centric sampling interest group that are developing different kinds of tubes and additives to hopefully stabilize samples. And we have a team at the moment, the patient-centric sampling interest group has a team at the moment developing further lists to go on our website of tubes and other technologies, containers that help stabilize and monitor samples. And hopefully in the next few months, those lists will be available on our website as well. So yeah, early days, and but I think there are use cases out there that might be easier to do. You don't want to start with an unstable molecule that has got a high chance of failure.
Mm. Okay, I think if there's no more questions, then we can wrap it up. Neil, thanks again for your presentation. I found it very insightful, and also quite a few people are commenting that it was insightful. I look forward to the presentations that's following. I'd also like to thank our audience for attending. Our next CPD event, Elna, is in 2 weeks, right? 2 weeks' time. So we will see you in 2 weeks. Have a good evening, and I wish you a night's rest. Thank you very much, and goodbye.
Thank you, everyone. Bye.